Hi guys, welcome to my channel. So today we will be learning how to design TETRA-ARMS primer for PCR. So for that you need to know which gene you are looking for and what is the SNP. So in my case it's CYP2C9. So I'll type it out in NCBI. Okay, and I can just select from here gene, or directly I can just go for all database. Anything is fine, it will give you the results. So once I click search, it will show me the sequence link to download. So since I need to know download it for humans, of course, so here you can see Homo sapiens and chromosome 10, everything is correct here. So I'll select it. Now as you can see, you need to just cross check the details of your selected gene. Now from here, download datasets, I click, it is already selected. As you can see, gene sequences FASTA. So I'll just click here on download, and you can see it's showing downloading, right? So it's downloaded now. So what I need to do is just open it and data gene. So as I open it, I'll get the complete gene sequence of CYP2C9 with all the other details, gene ID, chromosome number, everything. So you can see it is around quite big actually. So first part is done. So I'll just keep it as such. I'll minimize this. Now I'll go to again the browser. Now I want to know the sequence which is for which I want to design the primer. So in my case, I know what is the RSID. So I'll go to ensemble this time. I find it easy to use. You can use UCSC or other browser as well. So here, go to ensemble, click on human, because I'm trying to design it for human, of course. Click here on search, and this is my RSID. Click go. Ensemble is sometimes slow, so you can have patience. It's not necessarily that your internet is not working, right? So we have this page now. As you can see, all the details are given. You can just cross check missense variation and alleles, chromosome number, tag and all. So what I want now is I want the flanking sequence, right? I have clicked here. So as you can see, it is loaded here, and since I was already using this, so I'll show you one more thing. If you click here, show variants in flanking sequence, and click here, it will show you all the variants. One second, yeah, like this. But since we don't need all this, so we'll just again go to configure, click here, and that's it. So now we have a focus variant, and this is our SNP, basically what we are looking for. Now what we need to do is we need to check or maybe select this and find this same sequence in the downloaded gene sequence of CYP2C9, because that's a very big one. So it's very simple. Just click here and you can select this, control C, that is copy. Go to your downloaded uh, sequence, which is this, control F, and paste your sequence here. It will automatically search for it, and it has searched. So since you have seen, I had selected just before that red marked letter, so the next letter I'll select it and mark it, of course, red, right? So this is our SNP present basically, and the remaining sequence that I just now searched, selected, I'll mark it with any other color, maybe not yellow. Actually, I'll highlight it with pink, right? And same way, just to cross check that what we have downloaded is correct, I'll just go to 1 second. Yeah, so I'll just come here, select this, control copy, and again go to my document and just paste it in the search bar. Press enter. Since this uh, these letters are getting cut here, so of course it will not be showing me here. So what I have to do is I'll just delete one by one from this side. As you can see, when it comes for 1 2 3 4 around 6 7 bases, nucleotide bases, it will get highlighted, right? See, so like that it will be available on different places, but we need this one. So that is one confirmation that what we have selected is correct. Again I'll paste it and I'll try to delete it from this side. So if I delete it from this side means near to this red marked C, right? So if I delete it after 6 or 7 bases, it will highlight the next line. Yeah, so as you can see, this is now highlighted, right? So that means what we have selected is correct, and I'll highlight it with maybe some other color. So you can use press shift and just use the arrow keys to select one by one if you are not very well with the what do you say, mouse or the laptop pad. So I'll just keep it like that. So now these this pink one and this green one will be treated as your will be used for your designing the inner forward and inner reverse, right? So this pink side will be inner forward and the green side will be your inner reverse, right? So I'll leave it as such, and I'll again go to the different another software that we'll be using to design the primer. So this is what my software is, that is primer1.soton.ac.uk. You can note down the website. So here what you need to do, it's very simple. You can just make it in half. 1 second. Yeah, so here what you need to do is you have to randomly select a base basis below 1000, and making sure that this area where your SNP is located is also covered. So I'll just go maybe 3-4 lines above this, and I'll just select it, press shift and go down 1 2 3 like that. So I'll check how many characters I have selected, because the software won't take more than 1000 bases, it's clearly mentioned. So I'll click here and it says 840. Fine, so this is fine. So I'll just copy it, control C. Now I'll paste it in the software, control V, right? So now it is showing me some spaces, right? So I need to fill these gaps. Just place your cursor and click the press the delete button, and that's it. So it will take hardly 2 minutes. It happens sometime, not all the time. Okay, so I'll just check it. Okay, some more are there. My this SNP is in forward, is a forward strand, and some of you might have a reverse strand also, but doesn't matter, the designing will be done in the same way for forward and reverse as well. Okay, So this is done. Now it is asking me position of SNP from the start sequence. So from the start means from this T. Now how will I find it here? I have to just click here, okay, and I have to count till this C. So how to count this? Of course you can't do it manually. What you do is bring your cursor here, click it, and just press the shift, bring it down, okay. Now you can see it is selected. Now by the left arrow key you can bring it till this red C, right? You have to select it so that it gets covered, right? So now bring it here, your cursor, and click here. So it will show you characters, that is 413. So that is the location of the SNP, right? 413. So I'll type it in the software. Right now it is asking me the allele 1 and allele 2. So allele 1 is what is present here, and allele 2 is what it is getting replaced with. So where do I find it? I'll go to again ensemble, and C is replaced by A, right? So I'll go to the software again, type it out, or if you want to read about it, what is it, you can just click on the allele 1 and it will tell you what is it. So it is the SNP allele as indicated in the source sequence. That means it will be C, right? So we'll write here C. Capital, small, doesn't matter, it will take it as it is. So next is SNP allele 2 is the other allele that is not present in the source sequence. So for us we have A and T also, so I'll type A, right? So allele 2, I'll type A. Now I don't need to change anything else. Optimum primer size is 28, maximum is 30, minimum primer size I can reduce maybe to 20, okay, even 18 is also fine. Minimum 18 it should be, and maximum actually it is 25, but let it be like that. Optimum product size and all these things you can leave as such, since you might be learning, so you might not know how to change these and all, but this is fine, right? Maximum temperature is 80, minimum 50, like that, everything is okay. I'll click on pick primers, so it will show me the primers available. So I'll just put it on full screen, yeah. So as you can see, now it has shown me different primer sets. So this is one set, this is second, third, like that, 10 sets are there. So now from here I can select which primer I want. For example, forward inner primer, reverse inner primer, this is one set, okay. And same way the size is also given. So now what I need to do is I need to check which one is suitable for me. Of course I can take the primers directly from here, forward, inner, reverse, all these. So if for example I'll just do a quick check for you whether it is actually available in the original sequence that we downloaded. I'll just uh, click it twice and it gets selected. Control C, copy it and go to your file. Control F again and delete this and paste what you have copied from there, right? So now what you need to do is, of course I just now I told you that the since it is coming in second line, so it might not be taking it in one go, so you need to delete. Yeah, it has already searched. So you can see that it is found here, and if I just delete one more, you will see the changes in this area, okay, that is highlighted with pink, okay. That means the forward primer is correctly taken, right? So this way you can check. And reverse primer, you need to reverse this, I mean you can use this reverse complement. I'll just do it for you. Yeah, I have it in bookmark. So reverse complement, what I need to do is again double click here, control C, that means copy, okay, delete this, control V here, and I want a reverse complement, so just submit. Now this sequence, double click, control C, that means copy, and in your Word file you just control find, that means find it out. Okay, of course it will not be searching because of the same reason. I'll just zoom, make it full screen. Now you can start deleting one by one. Yeah, it is here. So as you can see, uh, when I delete, you can notice here, keep your focus here on the green part. As I am deleting this, it is getting deleted. So while reverse, I need to do the reverse complementation is because it is in reverse, so if I need to make it straight, then only I can find it in the original sequence, right? Same way, outer primer, if you want to check, just double click, control copy, and go to your Word file, control F, paste it here. Okay, so now I need to find it out. Where is it? 1, 2, yeah, here it is. So now since only half of it is highlighted, so first I'll just uh, highlight the available part, maybe with the light blue color. Remaining part I'll again paste it here, and I'll start deleting from this side. So it will select the remaining part as well. Right, it is here. So I'll click here, shift arrow button towards the left side, and same light blue color. So now this is my outer forward primer. Okay, then I'll go for my reverse, outer reverse, control copy here. Yeah, so since this is reverse, outer reverse primer, so again I need to reverse complement. So just I'll control copy, that means select it, go to reverse complementation, paste my sequence, submit, and from here control copy, selected. Now go to the word, in the navigation search, control V. Now I'll start deleting from the end. 1, 2, 3. So here it is highlighted, that means this is your outer reverse primer. I'll just highlight it with the same light blue color, and for the remaining sequence what I'll do is again I'll paste the same sequence and start deleting from this side. So of course too many will be deleted. Continue to delete. Since start showing too many regions where it is aligning, so you just need to check which is yours, or if you can remember, and just you can check it that way also. right? It is here. So these four that I deleted, these also I'll highlight, right? So now here you can see your this outer blue one is outer forward, this pink one is your inner forward, green marked in green is your inner reverse, and marked in this again light blue is your outer reverse primer. So this is how you have designed it. Now you need to check the specificity, how specific is this, because if it is not specific they will bind here and there and you will not get the proper results in your PCR, and you, especially when you run the gel, you will be getting several bands. So I'll make a separate video for how to check the specificity of the primers. Okay, so till then you can practice this and let me know in the comments if any doubts are there. Thank you so much.